All right, so today's PowerPoint will be on cell differentiation, which is the first part of the multicellular organisms, which is topic 3. So cells need to be able to specialize because an organism requires many different types of cells that all have specific purposes. And cells will all start out as what's called an undifferentiated cell, and that cell will then form different structures that allow them to have a specific function within the body. So if we have a look at our undifferentiated plant cell, that can differentiate into a root hair cell or a transpelt cell or a leaf cell. And all of those cells have different purposes. And it's the same with an undifferentiated animal cell that can then differentiate into a whole different types of cells that all have different purposes. So DNA is responsible for the specialization of a cell. And in order for us to understand how cell differentiation works, we first need to understand DNA, genes, and protein synthesis. So DNA is deoxyribonucleic acid, and it carries genetic information on its structure. DNA is usually located in the nucleus, and it's located or called chromatin, which is loosely condensed genetic material. This chromatin then condenses with the help of histones into what's called chromosomes just before cell division. So if we have a look at the structure of DNA, we have our double helix structure, what we talk about in the next slide. We then have our DNA that is wrapped around our histones, which is then condensed into what's called our chromatin, and then chromatin condenses into chromosomes. So DNA structure has two strands, and each strand has what we call bases that are called nucleotides. Now the nucleotides are joined together into what we call a backbone or a ladder type structure that has two backbones and our bases, and those bases are called nucleotides. It's then twisted into a form that looks like a double helix in the diagram here. And the way that these two strands of DNA hold together in this structure is that they have very weak hydrogen bonds in between the bases that allow them to join. So DNA on one strand of a sequence of DNA has what's called a genetic code. And that is all the information that the cell requires to be able to function properly, to perform all of its chemical processes, all the instructions. And a segment of these bases along one strand of DNA is what we call a gene. So, and genes code for proteins, and we need proteins to be able to perform some of our bodily functions, whether it be, for example, creating glucose or, breaking down glucose. So DNA will have segments of what's called introns, which are non-coding sequences of DNA, and exons, which are your coding sequences of DNA. So if we have a look at our DNA strand here, all of the bits that are, or the DNA strands that are highlighted in different colors called exons, they code for a specific protein, and each gene codes for a different protein. The introns are the other parts of the DNA strand that don't code for anything. Now, in order for us to get the instructions which is on the DNA to then make the protein, there are several instructions or steps that a cell must go through in order to create that protein. So the first step is transcription. So what happens there is we have our double-stranded piece of DNA and it unwinds in the place where we have our gene that's of interest to make the protein that we require. So what happens is we have the formation of what's called a messenger RNA strand, and this messenger RNA strand just pairs with the complementary base pairs on the template strand of DNA, and they form, with the help of RNA polymerase, they form what's called your messenger RNA molecule. Now, this messenger RNA molecule is the transcribed instructions, hence why it's called transcription. This is the instructions that we need to be able to make our protein. So the next step in protein synthesis, after we've transcribed the instructions, is the translation of the instructions. And this is where we translate that messenger RNA strand that we copied from the instructions on the DNA strand. We then take that out into the cytoplasm in the cell, and the messenger RNA will then assemble an amino acid chain called a polypeptide chain or a protein. So we can see that we've got these structures here, which are called transfer RNA, and they come in and they then help to build this amino acid chain here with our different amino acids.
DNA is very long and cumbersome. The strands of DNA also code for many different proteins. To get the code for the desired protein from the nucleus to the ribosomes in the cytoplasm, a copy of the small necessary section of DNA will be made. The copying process is called transcription, and it occurs in the nucleus. It's the first step in protein synthesis, the process of making protein. The second step is called translation. A portion of the DNA is unzipped so that the mRNA can be made from the DNA like a template. That's transcription. Nucleotides of RNA match with one strand of DNA and make mRNA. RNA polymerase unzips the DNA and puts RNA nucleotides into the right place. C and G nucleotides match up, A and T nucleotides match up, but anytime the RNA places a complement to an A on the DNA, it places a U instead of a T in the RNA. Remember, RNA uses uracil instead of thymine. So, if the DNA strand looked like this, the complementary mRNA strand would be the complement, but each time there would normally be a thymine, there's a uracil in its place. The mRNA also doesn't copy the entire strand of DNA. It only copies the portion of DNA that codes for the protein it wants at the time. In fact, most of DNA doesn't code for anything. Only about 2% of it codes for protein. So the small section of coding DNA has a start and stop signal. After the mRNA is formed, it moves out of the nucleus through nuclear pore and goes into the cytoplasm. Remember, there are 3 different types of RNA. The purpose of the mRNA is to get the protein code out of the nucleus without pulling the DNA out of the nucleus. Then the mRNA will need to find a ribosome in the cytoplasm. Ribosomes can be found on the rough ER or floating in the cytosol. mRNA is read 3 bases at a time, and these 3 bases are called codons. Now, the ribosomes are made of protein and RNA, and the ribosomes are the protein-making machines that read the mRNA code and add the correct amino acid using tRNA. Remember, tRNA stands for transfer RNA. And that's because its purpose is to transfer the right amino acid to the ribosome to build the protein. The tRNA has an anticodon on one end, which will match a specific codon on the mRNA, and has a specific amino acid on the other end. Together, these 3 parts will make translation happen. Translation occurs in the cytoplasm of the cell wherever there's a ribosome. To get translation started, mRNA attaches to a ribosome, and a start codon must be read. It's usually AUG, and I remember that school starts in August, and that helps me remember AUG. The first amino acid is brought in by tRNA. The anticodon on tRNA matches up to the codon on mRNA. Then the next tRNA molecule moves in and matches up with the mRNA codon. This time, the amino acids form a peptide bond and link together. Then, the first tRNA can detach, and the mRNA shifts through like ticker tape, and the next tRNA molecule can come in. The protein grows until a stop codon is reached. Once the stop codon is read, the protein is formed and ready to finish folding to become functional. And that's the end of protein synthesis. Now, we can figure out the sequence of amino acids using the mRNA and amino acid chart like this one. Let's use an example. Starting with the first 3 letters, AUG, we can find the amino acids. The first letter is A, which narrows it down to this row. The second letter is U, which narrows it down to this column, and the third letter is G, which means that this is methionine, which is the starting amino acid. Next is CCC, which is the second row, second column, and second row on the right. But we don't even have to look at the third letter in this case, because every third letter will bring the same amino acid, proline. Next is GGC, fourth row, fourth column, second row on the right. That gives us glycine. And lastly is UAA, row 1, column 3, row 3, and that gives us stop. There's no amino acid here. It just means that the protein will detach, and it marks the end of protein synthesis. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe, and follow me on Twitter at SciencePet.
Okay, so now that we understand how proteins need to be synthesized, we can then talk about cell differentiation. So as I've said before, all cells need to be able to differentiate, and they do so by producing different specific proteins that have specific purposes. Now, it's important to understand that all cells carry the same genes. All genes are the same, or all DNA is the same in each individual cell. However, what makes cells different is about which genes on that genetic material are activated, and this is called gene expression. So, gene expression occurs where specific proteins that are required in a cell, the genes that code for those proteins are switched on. And genes that are present but are not active, which means that they don't make those proteins, are called to be switched off. So multicellular organisms will switch genes on and off in several different ways depending on their internal and external environment, as well as being dictated by the requirements of the body. Now, there are several ways in which organisms can switch genes on and off. They can either block the RNA polymerase, which means that your translation won't occur, or they could stop the unzipping of the DNA, so you can't even form a messenger RNA. Or they could promote coiling of the histones that make the genes inaccessible. So as we looked at our structure of our DNA before, we saw coiling of the genes around the histones, and if that coiling becomes tighter, then the DNA can't even be accessed to make a template for translation. Now, the environment can also impact on gene expression as well. As I said in the last slide, some environmental factors will impact on which genes are expressed. So those factors could include diet, physical activity, as well as stress, and they can either determine whether a gene is expressed or silenced, which means it's either switched on or off. And what these changes in gene expression mean is that the phenotype of the organism will be different depending on which genes are expressed. So phenotype just refers to the physical structure of the organism, the appearance, so whether they have blue hair or black hair or long hair or curly hair, or their internal physiology as well, so whether they have all their functioning organs, for example.
Here's a conundrum. Identical twins originate from the same DNA, so how can they turn out so different, even in traits that have a significant genetic component? For instance, why might one twin get heart disease at 55 while her sister runs marathons in perfect health? Nature versus nurture has a lot to do with it, but a deeper related answer can be found within something called epigenetics. That's the study of how DNA interacts with the multitude of smaller molecules found within cells, which can activate and deactivate genes. If you think of DNA as a recipe book, those molecules are largely what determine what gets cooked when. They aren't making any conscious choices themselves. Rather, their presence and concentration within cells makes the difference. So how does that work? Genes in DNA are expressed when they're read and transcribed into RNA, which is translated into proteins by structures called ribosomes. And proteins are much of what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. The most common way interference happens is that DNA, or the proteins it's wrapped around, gets labeled with small chemical tags. The set of all of the chemical tags that are attached to the genome of a given cell is called the epigenome. Some of these, like a methyl group, inhibit gene expression by derailing the cellular transcription machinery or causing the DNA to coil more tightly, making it inaccessible. The gene is still there, but it's silent. Boosting transcription is essentially the opposite. Some chemical tags will unwind the DNA, making it easier to transcribe, which ramps up production of the associated protein. Epigenetic changes can survive cell division, which means that they could affect an organism for its entire life. Sometimes that's a good thing. Epigenetic changes are part of normal development. The cells in an embryo start with one master genome. As the cells divide, some genes are activated and others inhibited. Over time, through this epigenetic reprogramming, some cells develop into heart cells and others into liver cells. Each of the approximately 200 cell types in your body has essentially the same genome, but its own distinct epigenome. The epigenome also mediates a lifelong dialogue between genes and the environment. The chemical tags that turn genes on and off can be influenced by factors including diet, chemical exposure, and medication. The resulting epigenetic changes can eventually lead to disease if, for example, they turn off a gene that makes a tumor-suppressing protein. Environmentally induced epigenetic changes are part of the reason why genetically identical twins can grow up to have very different lives. As twins get older, their epigenomes diverge, affecting the way they age and their susceptibility to disease. Even social experiences can cause epigenetic changes. In one famous experiment, when mother rats weren't attentive enough to their pups, genes in the babies that help them manage stress were methylated and turned off. And it might not stop with that generation. Most epigenetic marks are erased when egg and sperm cells are formed. But now, researchers think that some of those imprints survive, passing those epigenetic traits on to the next generation. Your mother's or your father's experiences as a child or choices as adults could actually shape your own epigenome. But even though epigenetic changes are sticky, they're not necessarily permanent. A balanced lifestyle that includes a healthy diet, exercise, and avoiding exposure to contaminants may, in the long run, create a healthy epigenome. It's an exciting time to be studying this. Scientists are just beginning to understand how epigenetics could explain mechanisms of human development and aging, as well as the origins of cancer, heart disease, mental illness, addiction, and many other conditions. Meanwhile, new genome editing techniques are making it much easier to identify which epigenetic changes really matter for health and disease. Once we understand how our epigenome influences us, we might be able to influence it too.